Welcome to the webinar about Funguschain project, valorization of mushroom agro-waste to obtain high-value products. My name is Marta, and today I'll be the one explaining you about Funguschain. I'm going to introduce you to Funguschain project, its approach and methods, the products aimed for, the construction of the demonstration plant, and the sustainability issues. I'm going to explain you in more detail the different methods. Each year, more than 3 million tons of mushroom byproducts are generated in Europe. Imagine this completely filled with mushroom byproducts. Europe produces a huge amount of agricultural residues, also called agro-wastes, which are currently underutilized. We aim to use these underutilized mushroom agro-waste through cost-effective extractions to create new bio-based products. We do this at industrial level by using the waste from one of Monaghan Mushroom's farms. The project has received funding from BBIGU, a public-private partnership between the EU and the Bio-Based Industries Consortium. The consortium involves 16 partners from 10 different countries, 4 research and development entities, 4 large industries, and 8 small and medium enterprises. Strong scientific, technical, technological, and industrial skills are combined. The process designed for the valorization of the different biomolecules from mushroom wastes integrates well-established and scalable technologies from biomass biotechnology in a cascade approach. I'm going to explain you in more detail each step of this cascade approach. In particular, the cascade process takes advantage of cold water extraction to obtain fermentable sugars and proteins that could be separated through subsequent downstream processes, pressurized hot water extraction to obtain a glucan-rich extract and a residue rich in chitin glucan, which could be converted into chitosan by the acetylation and in simpler sugars by saccharification. Microwave-assisted extraction to isolate the lipophilic fraction rich in polyphenols and lipids. I'm going to explain you in more detail the different methods. Microwave-assisted extraction. Mushroom waste are first pre-treated to reduce the moisture of the samples and milled to reduce particle size and therefore enhance the mass transfer of the sequential extractive procedures. A first extraction will be carried out by MAE, microwave-assisted extraction. A dispersion of the powdered mushroom byproducts is loaded in the extraction chamber. Then, the microwave reactor rapidly heats up the dispersion. After a specific amount of time, the dispersion is unloaded, filtered, and further treated to obtain antioxidants, lipids, and polyols. These are applied later on in other processes for the cosmetic, food, and plastic sectors. Microwave-assisted extraction was proposed as the primary extraction technique, and their experimental parameters were optimized by using response surface methodology, RSM. Different parameters like time, temperature, microwave power, and ethanol-water ratio were studied and optimized using the total phenolic content, TPC, antioxidant activity, FRAP, and extraction yield as variables to be optimized. The main idea is to decrease the presence of ethanol in the solvent mixture to make costs more affordable. Nevertheless, it was observed that the best results were obtained at 50% mixtures of both solvents, while temperature was 80 degrees Celsius. The time was 5 minutes and 700 watts power was selected. The polyphenols profile was also determined by significant amounts of compounds with antioxidant performance in their composition. Pressurized hot water extraction. Mushroom biomass from Agaricus bisporus tissues have a very cross-linked and recalcitrant structure. which poses high technological challenges for the extraction and isolation of the constituent polysaccharides, mainly unfound beta-glucans, and proteins. Mushroom cut-offs has an initial composition of 10 to 15% protein in dry weight. In order to extract the valuable protein, polysaccharide and sugar components, we propose cascade process. First, we perform a cold water blending to isolate the soluble metabolites, in example mannitol, and proteins. Second, we subject the fungal biomass to a pressurized hot water extraction process for the recovery of soluble polysaccharides in polymeric form, mainly alpha and beta-glucans. Downstream, processing by membrane filtration can be applied to reach high purities of the valuable high molar mass glucans. Finally, we perform the deconstruction of the unextractable residue into fermentable sugars for the subsequent microbial production of biopolyesters. The proteins and polysaccharides can be directly used as food supplements with enhanced texturizing and probiotic properties. We have optimized the extraction conditions in terms of temperature, pH, and time in order to maximize total solid yields, composition, and molecular structure. In general, pressurized hot water extraction offers higher yields compared to traditional extraction. Tuning the process conditions promotes the selective isolation of the target alpha-glucan and beta-glucan extracts and enriches the chitin content in the residue. Saccharification and fermentation. Saccharification of the insoluble residue after pressurized hot water extraction enables the release of fermentable C6 sugars, glucose, for the subsequent fermentation into biopolyesters. The residue from the water extraction is saccarified to obtain fermentable sugars. The fermentable sugars are loaded in a reactor with a microorganism which is able to convert them into bioplastic. After a specific amount of time, the reactor is unloaded and the fermentation broth further treated to obtain a purified bioplastic powder. We have optimized the conditions for acid hydrolysis in terms of acid concentration, time, and temperature towards maximizing yields and minimal formation of inhibitors. We are also exploring routes for the enzymatic hydrolysis of the insoluble residue and compare this to chemical processes. Biopolyesters will be produced using the sugar platform to formulate novel bioplastic blends in compounding facilities for the plastic sector. Anaerobic digestion. The final residue is loaded in anaerobic fermentation reactors. The biogas produced by the microorganism during the anaerobic digestion is collected and used as fuel. We determine the biogas potential of several byproducts from the mushroom residue processing in a batch test as a first assessment of the suitability of these products in anaerobic digestion. Based on these results, continuous lab-scale anaerobic digestion reactors were started to simulate the process. This test provides us with the necessary information to design a full-scale anaerobic digestion plant and gives us information about the digestate characteristics. The data obtained in this work also fits the life cycle analysis, as recovering energy from residues is expected to improve the overall sustainability of the Funguschain treatment process. Let me explain you a bit more about our key products. These are the products that are under development. For food and nutraceutical sector, the intermediate products used are protein powder and glucans. We are trying to achieve natural, soy-free and lactose-free extracts to design a new food paste that could be shaped into a smooth bread or a chicken leg for the elderly nourishment. For the cosmetic sector, the intermediate products used are chitin residue and its derivative, and glucans. We are trying to achieve antioxidants, stabilizers, and preservatives for 100% natural cosmetic products. And for the bioplastic sector, the intermediate products used are fermentable sugars, glucans, and lipids. We are trying to achieve a compostable film for the cultivation of mushrooms, enhancing circularity of mushroom cultivation. Cosmetic products. Antioxidants extracted from the mushroom byproducts will be used in bio-based and environmentally friendly cosmetic products. The first step was to check if commercial chitin works as replacement for EDTA in cosmetics, and we have shown that it does. This has gained a lot of interest. Food products. Polysaccharides extracted from the mushroom byproducts will be used to supply dietary fibers while conveying the flavors and fragrances of mushrooms in nutraceutical products. The extract obtained, which contains antioxidant and antimicrobial additives, are being used to develop novel formulations for food applications for elderly people. These new formulations are fortified with natural antioxidants as well as protein-rich preparates. Product quality assurance analysis and shelf life tests are being carried out. We focus on cultivation residues-derived nutraceuticals to prevent obesity and combat metabolic syndrome. For this, we used assays representing central regulators of fat metabolism, the PPARs. Using extraction and separation methods coupled to bioanalysis, we have identified molecules that are responsible for PPAR activation. The purified fraction strongly suppresses fat cell differentiation and fat accumulation. We are now focusing on the identification of the most suitable ways to create active preparations as a basis for effective nutraceuticals. Bioplastic obtained from the fermentation of sugars will be used for the production of bio-based and biodegradable films. We have two streams in the bioplastic sector. On one hand, we have analyzed the residue of mushrooms in order to identify building blocks for preparing additives for lubricants and plastics. 4 products were identified, and those products are biodegradable and obtained by natural sources. Those products are blended with commercial biopolymers. Different combinations and amounts are prepared and tested at pilot scale. Once we have narrowed the amount of successful formulations, they are tested at industrial scale by fabricating bags and gloves. On the other side, tests are being carried out by introducing commercial PHB. The idea is that later on, that will be replaced by the one obtained in the project. We introduced the PHB in biopolymeric blends developed by one of our partners. These new formulations are being used for producing biodegradable match film. As I mentioned before, valuable compounds are extracted from the mushroom stocks, such as antioxidants, directly applied in plastics, and a sugar stream, which can be converted into PHB. These products can be used to make bioplastic products, but after their usage, they can be treated in an organic recycling process, such as composting. The obtained compost can be used for growing new biomass. Moreover, the goal is that the mulch film is biodegradable in soil. The spent compost, which is the growth substrate of the mushrooms, is currently discarded on the land after harvesting of the mushrooms. We are investigating if the spent compost can be used as a soil improver. It was observed that a small composting process is needed to stabilize the material in order to have good plant yields. Let's talk about the demonstration plant. The demonstration activities and biorefinery establishment have made a great progress. Monagat Mushrooms is constructing a demo plant in Ireland to demonstrate the process of generating a valuable product from mushroom in a cascading approach. The biorefinery will have an operational capacity of 5 tons a week of dried residues. That means a capacity of 78 tons a year. Feedstock preparation is fully installed, as well as mushroom powder production, thereby establishing the first step of the cascade, mushroom protein powder. Here are some pictures of different areas of the plant. These are pictures of processing equipment in cold water extraction and liquid-solid separation of fresh mushroom stalks. In the pictures, clockwise, you can see first the product intake, the stir tank and homogenizer, the slurred mushroom feed, insoluble intermediate residue, and liquid-solid separation through a GEA Westphalia decanter. Here, the downstream processing equipment, clockwise, ultrafiltration, transfer tanks, falling film evaporator, and spray drying. Now, let's go into sustainability. We have developed a life cycle analysis to evaluate the environmental sustainability of any system, extract, intermediate, and residue, obtained from the mushroom's waste valorization value chain, as well as any benchmark counterparts performing the same or a similar function. Environmental impacts are assessed from raw material extraction and processing through the product's manufacture, distribution, and use to their recycling or final disposal. The study involves the build-up of an inventory, input energy and materials as well as any emissions, of the product, process or service, and leads to the assessment of accumulative potential environmental impacts. Procedures for conducting LCAs are included in the 14,000 series of environmental management (ICO) standards of, and in particular, an ICO 14040 and ICO 14044. We are using human cell-based bioassays to measure potential toxicity and assure that products are safe. This test panel can rapidly evaluate major types of toxicity relevant for regulatory risk assessment and assurance of consumer confidence. Thank you for your attention. Now it's time for some questions. Please use the chat option to ask any questions.